Hello everyone. Today I will discuss the solutions of some questions taken from the chapter Biotechnology and its application from NCERT class 12 biology. Let's move to the first question. Crystals of Bt toxin produced by some bacteria do not kill the bacteria themselves because the options are given below. But before answering the question, we should know about what is Bt toxin. Well, the term Bt comes from the bacteria Bacillus thuringiensis. These bacteria produce a toxin that is named as Bt toxin, but the toxin is produced as its protoxin form, that is inactive form. When insect comes in contact with the toxin and ingests it, it goes to the gut of the insect, and there the alkaline pH changes it from its active form of toxin. When it changes to active form, it binds with the midgut epithelial of the cells and causes death of the insect. So it is all about Bt toxin. Let's see the question again. Now the options are: bacteria are resistant to the toxin, toxin is immature, toxin is inactive, bacteria encloses toxin in a special sac. So I have told earlier that it is the protoxin form, that is inactive. So the right answer is option C. Let's move to the next question, that is, what are transgenic bacteria? Illustrate using one example. Now transgenic bacteria are not any special type or some species or else. Rather, it is a bacteria that contains some foreign genes. Suppose the top left cell is a bacterial cell and the right side cell is a cell that contains some desired genes. So what we do, we just put our desired gene into the bacterial cell to make it transgenic. So we can define transgenic bacteria as: the transgenic bacteria is the bacteria that contains some desired foreign genes of our interest for the production of commercially important products. It may be medicines, it may be some dairy products or some other commercially important products. Now let us understand this with an example. Well, we all know about insulin, right? Insulin is a very important hormone for our human body that regulates our blood sugar level. Now think about a diabetic patient who needs a huge amount of insulin for daily basis. So from where would he collect this? It is obvious that he should collect it from some commercially, that is for medicine shops or else. At the past, we would collect the insulin from domestic cattle or pigs or etc. from the slaughterhouse. Now the insulin collected from the cattle are from foreign source, and we all know that foreign proteins can cause severe immune response to our immune system, and they may not be as efficient as our indigenous insulin. So the scientists took help of transgenic bacteria. Insulin has two chains, that is A chain and B chain. Now what scientists have do, they just put A chain in one Escherichia coli bacteria and B chain in another one. When the bacteria proliferates, they produce A chain and B chain separately in a large amount. Now it is very easy for the scientists to combine them with disulfide bonds and active insulin is produced. So you can see that active insulin is produced in a commercial manner in a large amount and very low time, it is commercially important. In 1983, an American company named Eli Lilly first produced such type of insulin from transgenic or genetically modified bacteria. Now let's move to the next question, that is compare and contrast the advantages and disadvantages of production of genetically modified crops. Now we all know that the genetically modified crops or organisms are very much advantageous to us. Does they have any disadvantages? Let's see. At first we take the advantages. Genetically modified crops are more tolerant to abiotic stresses. Some abiotic stresses like cold, too much hot or drought may cause severe losses to crops. Now some stress resistant genes are introduced into the crops that makes them tolerant to these abiotic stresses. Next point is less dependent on chemical pesticides. We all know that crops are very much attacked by insects, nematodes, etc. So we have to apply various chemical pesticides to protect them. These chemical pesticides are very harmful for the environment as well as human body. Now with the help of genetic engineering, it is possible to make transgenic crops that is resistant to some insects. Now the next point is to reduce post-harvest losses. When the crops are harvested and stored in storage houses, there may also attacks of rodents or insects may happen. If the crops may produce some indigenous products to protect themselves, it is very helpful, and it is done with the help of genetic engineering. The next point is more efficient mineral usage. These transgenic plants often use less amount of minerals from the soil, and it helps the next cycle or the next round of crop productions, and this prevents early exhaustion of fertility of the soil. And the most important point, enhanced nutritional quality of foods. Scientists have developed some crops of vitamin A producing, that is they introduced the gene for production of vitamin A into the crops that indigenously develops or produce vitamin A inside the crops. So we don't have to take it outside the crops in form of tonics or syrups or etc. It is very helpful for us. Now take us a look to the disadvantages. It can affect the beneficial insects. How? Think about the beneficial insects like the pollinators, wasps, bees, etc. affected with the toxin that is produced by the transgenic plant. If the pollinators are affected, then a huge loss in crop production may occur. The next point is the crops can affect the human health in long race. These transgenic crops produces a large amount of toxin to protect themselves from insects, but is the toxin safe for human beings? Well, this may be no, because toxins can be harmful for the human beings in long race. The toxin is stored in ecological succession, and it may harm the human beings also in further. The transgenic or genetically modified crops may endanger the native species, because we all know that native species play a very important role to maintain the natural biodiversity of a locality. if we introduce transgenic plants in a large amount, they may hamper the natural biodiversity of a location. Okay, now next to the next question, that is, what are cryoproteins? Name an organism that produces it. How has man exploited this protein to his benefit? Cryoproteins are mainly one type of toxin that is produced by cryogenes. Cryogenes may be of many types like Cry1AC, Cry1AB, Cry2AB, etc. These cryoproteins are produced by Bacillus thuringiensis bacteria that I have told earlier. Now this is a toxin, right? Man have exploited this toxin as his benefit also. They have produced Bt cotton, Bt corn, etc., which indigenously produce Bt toxin inside them to protect themselves from the attacks of insects, rodents, nematodes, etc. Now move to the next question, that is, what is gene therapy? Illustrate using the example of adenosine deaminase deficiency. Gene therapy is a technique for correcting defective genes in human beings as well as in other animals or plants. We all know that all the genes present in our genome is not active, or they may act wrongly, producing an offensive character. To rectify this, we may take help of gene therapy. So gene therapy mainly is a technique to rectify the defective genes or some deleted genes in human or plant body. Now let us know about with an example, that is ADA. ADA or adenosine deaminase is an important enzyme that is required for our normal functioning of immune system. With the deficiency of ADA, there develops a condition called severe combined immunodeficiency syndrome. That is a very difficult situation, and when the gene of adenosine deaminase is deleted somehow, or it is inactive in the body, then this deficiency occurs and this causes a severe disease. To rectify this, there are two processes. One is bone marrow transplantation and other is to give ADA enzyme injection to the affected patient, but these two are not permanent cure for the disease. So what scientists have done, they just took help of gene therapy. They took a bacteria or bacterial plasmid containing human ADA gene and an retroviral vector. When the bacteria proliferates, they produced plasmids or genes containing ADA gene. Now these genes are collected from them and are transferred to the T lymphocytes of the human body, or genetically. The gene therapy can help in the correction of adenosine deaminase coding gene. So this is all about I have to discuss today. See you later. Bye.